Welcome to another episode of Science Talks, a conversation hosted by the University of Arizona's BIO5 Institute. My name is Amy Randall Barber. Induced pluri-potent stem cells, or iPSC, are widely used in therapeutics for disease modeling, regenerative medicine, and drug discovery. Using patient cells, scientists can regenerate their cells into iPSC and recreate new organs for patients who need them. Today we are joined by Dr. Ekta Minocha, a 2023 BIO5 postdoctoral fellow working in the lab of Dr. Jason Wertheim here at the University of Arizona College of Medicine, Tucson. She is focused on developing bioartificial liver tissues. She hopes that one day they can replace a failing liver in the human body and expedite the waiting process for an organ transplant. Ekta, thank you so much for joining us today. We like to start every podcast the same way with a few fun icebreaker questions, rapid fire style, just completely unrelated to science. Question number 1. Do you have a hidden talent? And if so, what is it?
Yes, I like doing embroidery and I also like doing glass painting. I remember when I was in school during my summer vacations, I used to do embroidery on the cushion covers and pillow covers. I also gifted my grandma a handkerchief, which I embroidered myself.
Aww.
And I also gifted a few glass paintings to my friends and relatives.
That's
so
awesome.
I like doing that.
Do you still do it? Do you still do both?
Yes, if I get free time, I like doing glass painting.
Oh,
yeah.
Very nice. So I know with embroidery, there's tons of different things that you can actually design. What kind of designs do you make or do you like to make?
I like making flowers and doing putting mirrors with the threads on the cushion covers or pillow covers, like making them beautiful with colorful threads and different type of stitches.
Oh, very nice. All right. Next question. Who is your favorite Disney character and why?
Mickey Mouse is my favorite Disney character, and I like him for his cute smile, his innocence, and his kindness.
Oh, very nice. He's classic, right?
Yeah.
My daughter actually also loves Mickey Mouse. Would you rather travel to the past or the future?
I would like to travel in future because I am a curious person and I want to know what is going to happen in future.
Well, that makes complete sense to me. As a scientist, I would think that might help you with some of your work. So what was it that brought you to the University of Arizona and BIO5 Institute all the way from India, where you attended Sanjay Gandhi Postgraduate Institute of Medicine?
Since my school time, I've always been passionate about science, and biology has been one of my favorite subjects. And it really fascinates me, like how a single cell can give rise to a whole new individual. So I wanted to continue my passion in science. So I did my doctorate degree in the field of stem cells and tissue regeneration. And at the time when I was about to submit my thesis, I came to know about Dr. Jason Wertheim, and he was looking for a postdoc who had prior experience of working with stem cells and regenerative medicine.
Okay.
So I applied for that position and I was interviewed and I got selected.
Nice.
And it's really nice to be here. Like, people are really helpful. They are open for collaborations, and really exciting research is going on here, like at the University of Arizona and BIO5 Institute.
Well, that's always good to know.
Yeah.
That's good to hear, I should say. We know it, but it's good to actually hear stories about it or of it happening. Can you tell us about your overall research goals or the overall research goals of Dr. Wertheim's lab?
The overall research goal is actually to develop transplantable liver tissues that can repair or replace the failing organ, or they can shorten the time for transplantation. In this line, I'm working to generate liver organoids, which are like miniaturized organ in a dish that has similar function as that of the normal liver. And I'm using these organoids to model a disease in the liver called non-alcoholic steatohepatitis, and I'm also using that to study the underlying mechanisms of the disease.
Oh, wow, okay. Does Dr. Wertheim's lab only work on liver?
No,
tissues?
Dr. Wertheim's lab also works on kidney and also on bioprinting and developing bioartificial scaffolds.
Oh, okay. So can you tell us about your specific project in the lab and how you became how you came into that project?
My main project is developing liver organoids. So I'm using these organoids to model liver disease and called non-alcoholic steatohepatitis. And I'm also using them for understanding the underlying mechanisms, like how this disease is caused. And the long-term goal is to see whether we can transplant them in patients, or right now, like, I'm going to see whether these liver organoids have the potential to transplant in the animals to see their functionality and engraftment abilities.
Wow, cool. Can you tell us what challenges you've had during some of your experiments over your time in science?
Yes, like working with iPSCs is not easy. Everybody knows that it's challenging. We have to come to lab every day to change the media. Plus, iPSCs is a spoiled child, I should say.
Oh.
Yes, so they behave differently.
Oh.
Sometimes you do everything in a similar way, in a similar pattern, but next day you come to lab and you see all cells have died.
Oh.
There is no reason why we see that, but iPSCs behave in that way. So major challenge is that culturing iPSCs, working with iPSCs, and our starting material is iPSC. So we have to depend on them. They have to grow better so we can make the organoids and progress our research.
Yeah. Where do you get those from?
I get these iPSCs, some of them we have purchased from the CDI CERM stem cell repository. It's a stem cell bank in California. And some of them we have reprogrammed from patients with NASH. So we take the peripheral blood mononuclear cells of the NASH patients, or the patients who are undergoing liver transplantation. So we take the PBMCs, or peripheral blood mononuclear cells, and then we reprogram these PBMCs into iPSCs. So this work is done at Northwestern University. We have a collaborator there, Dr. Richard Green. So all this is taken care by him. So we get the stem cells here and we do the rest of the experimental work here at University of Arizona.
Oh, wow. You mentioned that Dr. Wertheim is a liver transplant, or I should say a transplant surgeon, but I was going to ask you, is he specifically liver, or does he do other organs?
He's a transplant surgeon. I'm not sure if he's involved into kidney transplantation as well, but I know about he's involved in liver transplantation.
Okay.
I'm not sure about kidney.
I'm just guessing that maybe that was part of why he started doing this type of work, seeing the weight that people had and their disease progressing during that weight and all of that. I don't know if he's shared any of that with you or if that's correct, but I would imagine.
No, I don't know much about his clinical part, but yes, in research, he's here with us every time. We can call him anytime to discuss any problem we are having. So he's there with us.
He sounds like a really great mentor.
Yeah.
Wonderful. You were a fellow in 2023, the BIO5 postdoctoral fellowship. And so can you tell us how you learned about the opportunity to become part of the fellowship and how the fellowship helped you with your project?
I came to know about this fellowship through my mentor. Dr. Jason Wertheim, he said me to apply for this grant, and I also got the email, so I applied for this grant. And the specific goals for this grant were to understand the interrelationship between the innate and environmental drivers of NASH. So I used iPSC derived hepatocytes, and I cultured them on different stiffnesses to study the effect of the environment on the behavior of those hepatocytes. Okay. So we were studying the relationship between the innate and environmental drivers, non-alcoholic steatohepatitis.
Okay, and did you get some results?
Yes. Of course, we saw that stiffness changed the lipid species, plus it also affected the gene expression levels of the genes which were controlling those lipid production and all.
Wow. And so you continue your work, and can you tell us how close are we to being able to use the tissues that you're developing for organ transplant in place of organ transplants?
Yes, it is still a long way to go. Like we are still in the preclinical trials right now. For going into the clinic, we need to do clinical trials and all. And just like a coin has two sides, the iPSCs also have benefits and also has limitations. Like they are really heterogeneous and there is batch to batch variation. So we need to overcome before that is going for transplantation in the patients.
What does that mean?
Like batch to batch variation, like sometimes these cells behave differently. Like sometimes, as you see in the liver, the major population is of the hepatocytes and other cells as well. But sometimes you see that non-parenchymal cell types, like other cell types, are more in the organoids as compared to the hepatocytes. So there is variation as you make the organoids from batch to batch.
Gotcha.
So we need to overcome that.
Yeah. Okay. That's
and there is lots of heterogeneity as well.
Well, thank you for doing that important work. Eventually you guys are going to nail this and you're going to be able to really heal a lot of people.
Hope we do that.
Yeah, absolutely. So do you have a mentor that has impacted your life?
Yes, of course. I have been really lucky to get great mentors in my life. Like my biggest mentors are my parents, who have always stood by me, supported me and motivated me. And I'm actually, due to them, that I am here. Like they have supported me in every sense. Plus, I was also lucky to get great mentors during my education period. Like some of them I should name, like Dr. Sonia Nityanand, Dr. C.P. Chaturvedi, Mr. Vinod Pandey, and Leena Chatterjee. And now here at the University of Arizona, Dr. Jason Wertheim is my mentor, and he's really motivating and inspiring, and it really helps me to improve myself with his guidance.
Yeah, well, and I can only imagine, being in your shoes, PhD, and Dr. Wertheim and his work, and then now you're working for him. I mean, that just must be so cool. His work is renowned, and he's a very brilliant, wonderful man. And so
he's a transplant surgeon.
Yeah, that is just super cool that you get to learn from him and work closely with him.
Yes.
So, do you think you'll stay at the University of Arizona? Do you think you'll go elsewhere?
I'm a J1 scholar, so after completing this duration, I have a 2-year home residency rule. So for that, I have to go to India for 2 years.
Okay.
So I will be looking for job positions in India, but I will apply for grants here as well, like during my period. But I plan to apply for faculty positions in India.
Oh, okay.
Yeah.
Okay. And do you already kind of know who you'd like to work with there? Or?
I will see, there are positions, so I will apply for them. So there are very limited positions available.
I would imagine this is a very specific type of work.
Yes.
Yeah. Well, I'm sure they'll be glad to have you and everything you've learned while you were here and to help their lab. So that's really it's just cool. I love all this stuff. I think it's just so amazing. So can you tell us what your why is? Sort of why you do what you do, what got you into it?
Actually, liver cancer, as you know, is associated with mortality, and only available therapeutic option available to the patient is transplantation. But again, transplantation has its limitation, like we need matched donor. So I'm working in this line. So our plan is to make transplantable liver tissues from the patient cells, so they are not rejected by the patient's body. So if this works in future, we will be able to develop transplantable tissues from the patient blood cells or skin cells. So this is why I'm working. So we don't have to wait for the transplant. We can shorten the transplant waiting time, and we can give other therapeutic options like liver organoids, and they can cure the disease.
Wow. Okay. And so this is for you work primarily with NASH,
yes.
The NASH liver disease. So can you explain to me, does that turn into cancer?
Yes. NASH is actually an intermediate stage between non-alcoholic fatty liver disease and cirrhosis. If NASH is not controlled, it may progress into cirrhosis, that is liver cancer, and ultimately the patient needs transplantation. Although NAFLT, which is the early stage, it is like reversible. But intermediate, I'm working on the intermediate stage to prevent it from moving into the cirrhotic stage. Like what are the mechanisms by which we can prevent it from progression to cirrhotic stage.
Okay. Well, thank you for explaining that. Wonderful. Well, thank you so much for being here with us today and telling us about your work.
Thank you for having me here.
Oh my goodness, absolutely. Is there anything else that you would like to share about your work or your time here at the university?
Yeah, like it has been great experience working with great collaborators. Like I have been collaborating with Dr. John Purdy. He's helping us out with the lipidomics work.
Okay.
And we are also working in like collaboration with like one of my mentors on the postdoc BIO5 grant was Dr. Nathan Sherrington.
Mm-hmm.
So he's like in the Department of Pharmacology. Like liver, as you know, is associated with drug metabolism. So he's helping us out with those transporters and drug metabolism studies. Other than that, we are also working on the microfluidics work to see the effect of flow on the organoids. So we are working with Dr. Zohar Yitzhak. So he is in the Aeronautical and Mechanical Engineering department. So yes, these are all the collaborators, and I really like, it's really fun being here. Like people are really helpful. They are very open. They are ready to collaborate. So it's really nice.
Well,
and science is all about collaborations.
Right. I was just, well, and BIO5 is all about collaboration. And it sounds to me like you are like the poster child for BIO5 because you're collaborating, across disciplines, just like what we promote. And it's just a real life example of that.
And I should say that Dr. Wertheim always promotes for collaboration. Like he's always open for collaborations.
Nice.
Yeah. And he always promotes me to take leadership in our collaborative meetings and everything, like to explain my work and everything. Like he always promotes me for taking the leadership role.
He sounds like a really good mentor.
Yes.
That's awesome. That's wonderful. Well, thank you again for being here. We really appreciate your time and really appreciate you sharing your information about your project and your story.
Thanks to our listeners for tuning in to another episode of Science Talks. Continue the conversation with us next time as we learn more about the amazing science happening at the University of Arizona's BIO5 Institute.